Hi everyone. Welcome. If you are here right now, it might be because the day has been long, and the night feels like a good time to slow down, to breathe a little more quietly, and to let your mind drift somewhere calm and interesting. Tonight, we're going to spend some time together, thinking about one of the most gentle and quietly fascinating subjects in all of science. We're going to think about bacteria. Now, bacteria might not sound like the most relaxing topic at first, but when you really take a moment to sit with it, when you let your mind settle into the quietness of it, you begin to realize that the world of bacteria is one of the most peaceful, ancient, and quietly remarkable things that exists. Bacteria have been here far, far longer than we have. They were here before the oceans took their current shape. They were here before plants learned to grow leaves. They have seen billions of years pass by, slowly, quietly, doing the same gentle things they have always done. And we can take a moment to notice that there is something deeply comforting about that, about things that simply continue, that quietly persist, that do not rush. So let your body relax, let your breathing slow just a little, and let us spend the next little while together, wandering softly through the quiet and beautiful world of bacteria. There is no need to rush. We have all the time we need. Bacteria are among the oldest living things on Earth, and we can take a moment to let that idea settle softly in the mind. The earliest known fossil evidence of bacteria dates back to approximately 3.5 billion years ago. That is a number so large it is almost dreamlike to think about. For reference, the Earth itself is about 4.5 billion years old. So bacteria appeared relatively early, not long after the planet had cooled enough to allow liquid water to exist on its surface. For most of Earth's history, for most of the time this planet has existed, bacteria were the only life here. They drifted through ancient oceans. They settled on bare rocks. They breathed and grew and quietly multiplied. In a world with no animals, no plants, no voices, no footsteps, just the slow turning of the planet, and the gentle work of these tiny, ancient beings. Let us think quietly about just how small bacteria are, because their smallness is one of the most gently astonishing things about them. A single bacterium is typically somewhere between 1 and 10 micrometers in length. A micrometer is 1/1,000,000th of a meter. To help imagine this, if you placed about 1,000 bacteria end to end in a row, that row would be roughly the width of a single human hair. And a human hair is already something we consider quite delicate and thin. So bacteria exist in a world of almost incomprehensible smallness. They live in the spaces between things we can barely see. And yet, within that tiny scale, they carry out all the same fundamental processes of life. They grow. They respond to their environment. They reproduce. Slowly, gently, they do everything that life requires, in a space too small for any human eye to observe directly. Gently moving forward, let us think about where bacteria actually live. And the answer, quietly, is almost everywhere. Bacteria have been found in the deepest ocean trenches, where the pressure is crushing and the darkness is total. They have been found in the ice of Antarctica, frozen for thousands of years, still alive. They have been found in the boiling water of hydrothermal vents, where temperatures exceed the normal boiling point of water. They have been found floating in the upper atmosphere, drifting gently through clouds. They have been found deep underground, kilometers below the surface, living in the slow warmth of the Earth's crust. Bacteria live in soil, in rivers, in the air we breathe, in our food, in our bodies. There is almost no environment on Earth where bacteria have not found a way to live quietly and persistently. They are, in the gentlest possible sense, everywhere. Now, as we continue, Let us take a quiet moment to think about the sheer number of bacteria in the world. Scientists have estimated that the total number of bacteria on Earth is somewhere around 10 to the power of 30. That is a 1 followed by 30 zeros. It is a number so vast it cannot really be visualized. For comparison, the number of stars in the observable universe is estimated to be around 10 to the power of 24. So there are, quietly and without any drama, more bacteria on Earth than there are stars visible in the entire known universe. And we can take a moment to notice how strange and soft and wonderful that is. That right now, in the soil outside, in the water in your glass, in the very air of the room you are in, there are more living things present than there are stars in the sky. Slowly, gently, let us think about the weight of all those bacteria. Scientists who study the distribution of life on Earth have calculated the total biomass of all living things. And when they do, bacteria account for an enormous portion of it. It is estimated that the total carbon mass of all bacteria on Earth is somewhere around 70 gigatons of carbon. For comparison, all of humanity combined, every person on the planet, accounts for roughly 0.06 gigatons of carbon. So in terms of the sheer biological weight of life on Earth, bacteria are overwhelmingly, quietly dominant. They are not the loudest form of life. They make no sound. They build no cities. And yet in terms of mass, of number, of age, and of reach, they are far and away the most significant living things this planet has ever produced. Let us take a quiet moment to think about the shapes that bacteria can take, because there is a kind of quiet elegance in bacterial geometry. Most bacteria fall into a few basic shapes. There are spherical bacteria, called cocci, which are small and round like tiny gentle beads. There are rod-shaped bacteria, called bacilli, long and straight or slightly curved, like miniature pillows. There are spiral-shaped bacteria, called spirilla, which wind gently through liquid like tiny corkscrews moving in slow motion. And there are comma-shaped bacteria, called vibrio, which curve softly like a question mark. Each shape has its own quiet advantages. Spherical bacteria can pack together efficiently. Rod shapes allow for more surface area. Spiral shapes help bacteria move through thick or viscous environments. Slowly, gently, the shape of each bacterium is a kind of quiet adaptation, a soft solution to the challenges of surviving in a particular world. Now, as we continue thinking about bacterial shapes, there is something particularly lovely about the way some bacteria arrange themselves in groups. When spherical bacteria divide and stay attached to each other, they form soft clusters or long chains. Bacteria that form clusters like bunches of grapes are called staphylococci, and bacteria that form long, bead-like chains are called streptococci. These names sound complicated, but the images behind them are actually quite gentle and beautiful when you visualize them. Imagine tiny pearls, strung together in a soft line, or tiny spheres clustered loosely together like a small bunch of grapes. These arrangements happen simply because after division, some bacteria do not separate fully. They remain connected, and so they grow in these soft, organized patterns, not because they plan to, but simply because that is what their nature quietly leads them to do. Gently moving forward, let us think about the inside of a bacterium, compared to the cells in our own bodies. A bacterial cell is beautifully simple. Human cells and animal cells are what scientists call eukaryotic, meaning they have a nucleus, a protected inner chamber where the genetic material is stored. Bacteria do not have a nucleus. Their genetic material, their DNA, floats freely within the cell in a region called the nucleoid. There are no internal walls, No separate compartments the way our cells have. Just a single, quiet space containing everything the bacterium needs to live. And we can take a moment to notice how elegant that is. A single tiny room. Everything needed for life. Present in one open, unpartitioned space. No separation. No hierarchy. Just the quiet wholeness of a simple, complete system. Let us think quietly about the outer covering of a bacterium. Because the bacterial cell wall is one of the most important and softly fascinating structures in all of biology. Most bacteria have a cell wall that surrounds them, just outside the cell membrane. This wall gives the bacterium its shape and protects it from the outside world. The material that most bacterial cell walls are made of is called peptidoglycan. A long, cross-linked mesh of sugar and protein molecules that forms a kind of soft, flexible armor around the cell. This wall is strong enough to maintain shape under pressure, but flexible enough to allow the cell to grow and divide. There is no need to rush through this idea. Just let it settle gently. A tiny, ancient being, wrapped in a wall made of woven sugar and protein, quietly maintaining its shape in a vast and sometimes harsh world. Slowly, gently, some bacteria have an additional layer beyond their cell wall called a capsule. The capsule is a loose, gel-like coating that surrounds the entire bacterium like a soft cloud. It is made mostly of polysaccharides, long chains of sugar molecules. The capsule serves several quiet purposes. It helps some bacteria stick to surfaces, anchoring them softly in place. It helps some bacteria resist drying out, holding a gentle layer of moisture around themselves. And in some cases, it helps bacteria avoid being detected or destroyed by other organisms. The capsule is one of those beautifully understated structures in biology. It does not flash or announce itself. It simply exists, a quiet, gentle layer between the bacterium and the world, offering a kind of soft protection that is easy to overlook but deeply important to the life within. Now, as we continue, let us think about how bacteria move. Some bacteria are non-modal, meaning they do not move on their own at all. They are simply carried by water currents, or by other organisms, or by the movement of air. But many bacteria are able to move quite actively. And the way they do it is one of the most quietly remarkable things in all of biology. Many bacteria move using structures called flagella. A flagellum is a long, thin, whip-like tail that extends from the body of the bacterium. By rotating this tail, literally spinning it like a tiny propeller, the bacterium can move through liquid. And we can take a moment to notice how extraordinary that is. A bacterium, with no muscles, no nervous system, no brain, rotates a molecular propeller to swim through its environment. Life finding a way to move through the world. In the simplest, most elegant way imaginable. Let us take a quiet moment to think about the bacterial flagellum more carefully, because it is genuinely one of the most intricate structures produced by life at the microscopic scale. The flagellum is not simply a rigid tail. It is a complex molecular machine, assembled from dozens of different protein components. At its base, where it connects to the cell, there is a structure called the basal body, a set of rings that anchors the flagellum to the cell wall and membrane. This basal body functions like a rotary motor. It spins. It literally rotates, powered by a flow of ions across the cell membrane. The flagellum can spin at speeds of up to 1,000 revolutions per minute. Slowly, gently, think about that. A molecular motor, smaller than anything human hands could build, spinning quietly inside a living cell. Biology at its most quietly astonishing, gently moving forward. Some bacteria move in a completely different way, without flagella at all. Some bacteria glide. Gliding bacteria move smoothly across solid surfaces. Not swimming, not rolling, but sliding with a slow, continuous, almost meditative motion. The exact mechanism of gliding varies between different types of bacteria. Some appear to use tiny thread-like structures called pili, which extend forward, attach to the surface, and then retract, pulling the cell gently forward. Others seem to secrete slippery molecules underneath themselves that allow them to slide. And some appear to use wave-like movements in their outer membrane. Whatever the mechanism, gliding bacteria move with a kind of quiet grace. They do not swim through liquid. They simply slide, slowly and persistently across whatever surface they happen to be resting on. Now, as we continue, let us think about how bacteria know which direction to move. Even though bacteria have no brain, no nervous system, and no sensory organs in any familiar sense, they are able to sense their environment and move toward or away from certain things. This ability is called chemotaxis. Bacteria can detect the presence of chemicals in their surroundings. Nutrients, for example, or potentially harmful substances. When a bacterium detects a source of nutrients, it biases its movement toward that source. When it detects something harmful, it biases its movement away. This is not thinking. It is not decision-making in any conscious sense. It is a purely chemical process, the bacterium's internal chemistry responding to signals in the environment. And yet the result, a tiny living thing navigating toward food and away from danger, feels almost gentle and purposeful when you observe it. Slowly, gently, let us think about another form of bacterial movement called twitching motility. Some bacteria move using very short, jerky movements, extending and retracting tiny protein filaments called type 4 pili. These pili reach out, attach to a surface, and then retract, pulling the bacterium forward in a small, twitching motion. It is not smooth or graceful in the way that gliding is. It is more like a slow, patient series of tiny pulls. Over time, though, this method of movement can carry bacteria across surfaces with surprising efficiency. Twitching motility is used by some bacteria to form biofilms, which we will talk about later in our time together. For now, just let the image sit softly in your mind. A tiny living thing, reaching out, gripping, pulling itself forward, slowly, patiently, one small movement at a time. Let us take a quiet moment to think about how bacteria get their energy. In our own bodies, we eat food, digest it, and use the energy stored in its molecules to power everything we do. Bacteria do something similar, but the variety of energy sources they can use is almost breathtakingly wide. Some bacteria, like us, get their energy by consuming organic molecules. Sugars, proteins, fats, these are called heterotrophs. But other bacteria can get their energy from completely inorganic sources. From minerals, from hydrogen gas, from iron compounds, from ammonia, and some bacteria can get their energy directly from sunlight, through a process like photosynthesis. This range of metabolic strategies means that bacteria can find energy almost anywhere on Earth, where other forms of life would starve. Some bacterium has quietly evolved a way to extract energy from whatever is available, gently moving forward. Let us think about photosynthetic bacteria, because they have a deeply important and gentle place in the history of life on Earth. Long before plants existed, long before the first leaf reached toward the sun, certain bacteria were already capturing sunlight and using it to make food. The most important of these were the cyanobacteria. Cyanobacteria are blue-green bacteria that evolved the ability to perform oxygenic photosynthesis, which means that as a byproduct of capturing sunlight, they produce oxygen, and for billions of years, quietly, without any awareness of what they were doing, cyanobacteria released oxygen into Earth's ancient atmosphere. Over time, this slowly transformed the planet's air. The oxygen we breathe today, the oxygen that makes complex life possible, is largely the accumulated product of billions of years of quiet bacterial work. Now, as we continue, let us think about bacteria that breathe in ways very different from our own. We breathe oxygen. We take it in, use it to burn fuel molecules, and release carbon dioxide. But many bacteria do not use oxygen at all. They are anaerobic, meaning they live and thrive in environments without oxygen. Some anaerobic bacteria actually find oxygen harmful, even deadly. They live in places where oxygen cannot reach. Deep in soil, in the gut, in sediments at the bottom of lakes. These bacteria use other molecules in place of oxygen. Sulfate, nitrate, carbon dioxide, as the final acceptor in their energy-producing reactions. The end products of their metabolism are often things like hydrogen sulfide, methane, or nitrogen gas, and we can take a moment to notice how quietly remarkable it is that life can run on so many different chemical fuels. Let us take a quiet moment to think about bacteria that eat rocks. This sounds dramatic, but it is actually one of the gentler and more slowly paced aspects of bacterial life. Certain bacteria are able to extract energy from inorganic minerals, iron compounds, sulfur compounds, manganese. These bacteria are called lithotrophs, which simply means rock eaters. They live in environments like deep mine shafts, ocean floors, and rocky soils, places where organic food is scarce or absent. Slowly, over long periods of time, these bacteria dissolve and transform the minerals around them. They play a quiet but important role in the cycling of elements through the Earth's crust. Iron, sulfur, and manganese, all slowly moved and transformed by the patient, unhurried work of tiny bacteria that have been doing this, in one form or another, for billions of years. Slowly, gently, let us think about the process by which bacteria break down organic matter. When a leaf falls from a tree and lands on the forest floor, when a piece of food is left out too long, when an animal dies in the wild, bacteria are among the first and most important decomposers at work. Bacteria secrete enzymes into their environment, proteins that break down complex organic molecules into simpler ones. These simpler molecules can then be absorbed by the bacterium and used for energy. This process of decomposition is not only necessary for bacterial survival, it is essential for the entire cycle of nutrients on Earth. Without bacteria to break down dead organic matter and return nutrients to the soil, the cycle of life would slow and eventually stop. In the quietest, most fundamental way, bacteria are the recyclers of the living world. Now, as we continue, let us think about how bacteria reproduce. The most common method of bacterial reproduction is called binary fission. It is a beautifully simple process. A bacterium grows to a certain size. It copies its DNA. The two copies of DNA move to opposite ends of the cell. Then the cell slowly pinches in the middle and divides into two identical daughter cells. Each daughter cell is a complete, functional bacterium, carrying its own copy of the genetic information. No partner is needed, no complex process of fertilization, just one cell, quietly becoming two, and each of those two cells can, under the right conditions, do the same thing again. There is something deeply restful about the simplicity of it. 1 becomes 2. 2 become 4. 4 become 8. Slowly, steadily, without drama or complexity. Let us take a quiet moment to think about how quickly some bacteria can reproduce. Under ideal conditions, with the right temperature, the right nutrients, the right level of moisture, some bacteria can divide as frequently as once every 20 minutes. That means that in 1 hour, a single bacterium could theoretically become 8 bacteria. In 2 hours, 512. In 3 hours, nearly 33,000. In theory, if conditions remained perfect and nothing interfered, a single bacterium could produce a colony of millions within a single day. Of course, in the real world, conditions are never ideal for long. Nutrients run out, space becomes limited, temperature changes, other organisms compete, and so bacterial populations do not actually grow without limit. But the potential, the quiet, patient potential for rapid growth, is always there, waiting softly for the right conditions, gently moving forward. Let us think about what happens to bacterial growth when conditions are not ideal. Scientists studying bacterial growth have described a gentle pattern called the bacterial growth curve. When bacteria are first introduced to a new environment, they do not immediately begin dividing rapidly. There is first a period called the lag phase, a quiet time of adjustment, during which the bacteria are adapting to their new surroundings, synthesizing the enzymes they need, and preparing for growth. This lag phase can last minutes or hours, depending on the conditions. It is a period of quiet readiness. The bacteria are not dormant. They are active, preparing, and we can take a moment to notice how gentle and natural that is. Before the work begins, there is always a period of quiet preparation. Now, as we continue, after the lag phase comes the exponential phase, also called the log phase. This is when bacteria begin dividing rapidly, doubling at a regular interval. It is the period of most active growth. But this phase cannot continue forever. As bacteria consume nutrients, nutrients become scarce. As they produce waste products, those products accumulate. The environment slowly becomes less hospitable, and so the growth rate begins to slow. This transition marks the beginning of what is called the stationary phase, a quiet plateau where the rate of new bacteria being produced equals the rate of bacteria dying. The population holds relatively steady, and then, if conditions continue to worsen, comes the decline phase, a slow, gradual decrease in population as death outpaces growth. A full cycle: growth, plateau, and quiet decline, a gentle rhythm of life. Slowly, gently, let us think about a remarkable survival strategy that some bacteria use when conditions become harsh. Certain bacteria, most notably the genus Bacillus, are able to form structures called endospores. When a bacterium senses that conditions are becoming unfavorable, that nutrients are running low, or that temperature or moisture is changing, it begins the process of forming an endospore. The bacterium essentially packages its genetic material and essential proteins into a tough, protective shell within its own body. This endospore is extraordinarily durable. It can survive extreme heat, extreme cold, radiation, and the complete absence of water or nutrients. The bacterium, in a sense, goes to sleep, not a light sleep, but a deep and almost timeless dormancy. Endospores have been revived after being dormant for thousands, and possibly millions of years, a quiet waiting that outlasts almost everything. Now, as we continue, let us take a quiet moment to think about biofilms. Most of the time, when we think about bacteria, We imagine them as individual, free-floating cells. But in nature, many bacteria prefer to live in communities, attached to surfaces and to each other, in structures called biofilms. A biofilm begins when a few bacteria settle onto a surface, a rock in a stream, a tooth, a pipe, and attach themselves. They begin to multiply, and as they do, they also secrete a matrix of sticky molecules around themselves. Polysaccharides, proteins, and DNA. This matrix holds the community together like a soft, biological glue. Over time, the biofilm grows and develops structure. It is not just a random pile of bacteria. It is an organized community, with channels for water flow, with different bacteria performing different functions in different regions. A quiet city of microscopic life. Let us think gently about the protective benefits of living in a biofilm. Bacteria within a biofilm are considerably more protected than free-floating bacteria. The surrounding matrix acts as a physical barrier, slowing the penetration of harmful substances. Bacteria deep within a biofilm may be in a kind of slow, low-activity state, almost like a gentle sleep, which makes them less vulnerable to certain kinds of damage. The community nature of the biofilm also allows bacteria to share resources and to collectively resist environmental stress. There is something quietly beautiful about the idea of bacteria finding safety in community, in proximity to one another, in the gentle shelter of a shared structure. Individual cells that would be vulnerable alone become part of something more resilient and persistent together. A soft reminder that community offers a kind of protection that solitude cannot. Gently moving forward, bacteria within communities communicate with each other using a process called quorum sensing. This is one of the most softly fascinating aspects of bacterial social life. Bacteria constantly release small chemical signal molecules into their environment. As the bacterial population grows and becomes more dense, the concentration of these signal molecules increases. When the concentration reaches a certain threshold, a quorum, in a sense, bacteria throughout the community begin to change their behavior in response. They might start forming a biofilm. They might start producing certain proteins. They might coordinate a shared response to a threat, all without any central brain or command system. Just chemistry, just concentration, just quiet chemical signals accumulating in the space between cells, until a threshold is reached, and the community quietly shifts its behavior together. Now, as we continue, let us think about how bacteria share genetic information with each other. In our own biology, genetic information passes from parent to child through reproduction. But bacteria have evolved additional ways to exchange genetic material, even between unrelated individuals. One of the most important of these is called conjugation. In conjugation, two bacteria come into direct contact. One bacterium extends a thin protein tube, called a pilus, toward the other, drawing the two cells close together. A connection forms between them, and through this connection, a small piece of circular DNA, called a plasmid, can pass from one bacterium to the other. The receiving bacterium now carries new genetic information that it did not have before. This is one of the ways that genetic adaptations, including resistance to antibiotics, can spread through bacterial populations, quietly, through contact, through connection, slowly, gently. There is another way bacteria can acquire new genetic information, through a process called transformation. In transformation, a bacterium simply takes up free DNA from its environment. When bacteria die and their cells break open, their DNA is released into the surrounding liquid. Some bacteria have the ability to absorb fragments of this free-floating DNA and incorporate it into their own genome. This process was actually one of the first pieces of evidence that DNA is the carrier of genetic information. In the 1920s and 1940s, scientists observed that non-harmful bacteria could be transformed into harmful ones simply by being exposed to DNA from dead harmful bacteria. It was a quiet, gentle experiment, bacteria absorbing information from their environment, that helped unlock one of the fundamental secrets of life. Let us take a quiet moment to think about the bacteria that live within and on our own bodies. This community of bacteria is called the human microbiome, and the number of bacteria living in and on a single human body is genuinely staggering in the softest possible way. Estimates suggest that there are approximately 38 trillion bacterial cells in and on the human body, and the total number of human cells in the body is roughly 30 trillion. So in terms of cell count, there are slightly more bacterial cells in your body than there are human cells. And we can take a moment to notice how gently that reshapes our sense of what we are. We are not simply individual organisms. We are walking ecosystems. We are hosts to an enormous, quiet, living community that is in many ways as much a part of us as our own cells. Now, as we continue, the largest and most complex community of bacteria in the human body lives in the gut. The gut microbiome, the collection of bacteria, and also some other microorganisms living in the digestive system, is estimated to contain somewhere between 500 and 1,000 different species of bacteria. These bacteria perform an enormous range of functions that are quietly essential to our health. They help digest certain types of food that our own digestive enzymes cannot break down, particularly certain plant fibers. They produce vitamins, including vitamin K and several B vitamins, that we absorb and use. They help train and regulate our immune system. They produce chemical signals that travel through the bloodstream and may influence our mood and brain function. A quiet community, deep within us, gently doing more than most of us ever realize, gently moving forward. Let us think about the bacteria that live on our skin. The skin microbiome is a complex community of bacteria adapted to the relatively dry, salty, acidic environment of the skin's surface. Different regions of the skin support different communities of bacteria. The moist folds of the body, the armpits, the inside of the elbows, between the toes, are home to bacteria that prefer wetter environments. The relatively dry, exposed surfaces of the arms and legs support a different community. The face, with its abundance of oil-producing glands, supports yet another group. This quiet patchwork of bacterial communities across the surface of the skin is thought to play an important role in protecting against harmful bacteria. By occupying the skin's surface, these gentle resident bacteria make it harder for potentially harmful newcomers to establish themselves. Let us take a quiet moment to think about how we acquire our first bacteria. When we are growing in the womb, we are largely sterile, surrounded by the protective environment of the womb with very little bacterial exposure. But from the moment of birth, bacteria begin to colonize us. Babies born through vaginal delivery are colonized first by bacteria from the mother's vaginal and intestinal microbiome. Babies born by cesarean section acquire their initial bacteria from the environment and from skin contact. In the first days, weeks, and months of life, with every breath, every touch, every swallow of milk, a baby's microbiome develops and diversifies. The bacteria that colonize us in early life help shape our immune system, our metabolism, and possibly even aspects of our neurological development. Those first quiet encounters with bacteria set the foundation for a lifetime of gentle coexistence. Slowly, gently, let us think about the relationship between bacteria and our immune system. The immune system does not simply try to destroy all bacteria it encounters. It has learned, over evolutionary time, to distinguish between the bacteria that belong, the residents of the microbiome, and bacteria that are potentially harmful. This distinction is not always perfect, and sometimes goes wrong, but for the most part, our immune system has developed a kind of quiet tolerance for our resident bacteria. And more than tolerance, our immune system actually needs those bacteria to develop properly. Research in animals raised in completely sterile environments, with no bacteria at all, shows that without bacterial exposure, the immune system does not develop normally. It is as though the bacteria teach our immune cells what normal looks like. They help calibrate the system, gently, quietly, from the very beginning. Now, as we continue, let us think about one of the most important roles that bacteria play in the natural world: nitrogen fixation. Nitrogen is an essential element for all living things. It is a key component of proteins and DNA. The atmosphere is about 78% nitrogen gas. But in its gaseous form, nitrogen is unavailable to most plants and animals. It needs to be converted, fixed, into a form that living things can use. Like ammonia or nitrate, and certain bacteria, called nitrogen-fixing bacteria, are the primary agents of this conversion. Some of these bacteria live freely in the soil. Others live in intimate partnership within the root nodules of legume plants like peas and beans. These bacteria quietly take nitrogen from the air and convert it into a form that plants can absorb. Without this quiet bacterial work, the nitrogen cycle, and therefore most of life on Earth, would not function. Let us take a quiet moment to think about bacteria and the carbon cycle. Carbon moves through the living world in a slow, continuous cycle. Plants absorb carbon dioxide and build it into organic molecules. Animals eat those plants. When organisms die, their carbon-rich remains are broken down, and bacteria are central to this breakdown process. Decomposing bacteria break down dead organic matter, releasing the carbon it contains back into the environment as carbon dioxide, or incorporating it into the soil as organic material. This process is happening constantly, quietly, in every field and forest and ocean on Earth. In the deep sea, where organic matter drifts down from above and settles on the ocean floor, bacteria break it down slowly in the cold and dark. In the warm, moist soil of a rainforest, bacteria work quickly and continuously, always recycling, always returning, always completing the gentle circle, gently moving forward. Let us think about bacteria that live in extreme environments. Scientists use the term extremophile to describe organisms that thrive in conditions that would be lethal to most life. And bacteria are among the most accomplished extremophiles on Earth. Bacteria called thermophiles live in hot springs and hydrothermal vents at temperatures above 80 degrees Celsius. Bacteria called psychrophiles live in ice and permafrost at temperatures well below zero. Bacteria called acidophiles live in environments so acidic they would dissolve many materials. Bacteria called halophiles thrive in environments far salter than the ocean. The existence of bacteria in all these environments tells us something quietly profound about the nature of life. Life is not fragile. Life is not easily stopped. It finds a way, quietly and persistently, into almost every corner of the possible. Now, as we continue, let us think about the relationship between bacteria and plants. Most plants depend on bacteria not just for nitrogen, but in many other ways. The soil around plant roots, a region called the rhizosphere, is home to a rich and complex community of bacteria. These bacteria help break down organic matter in the soil, making nutrients available to plant roots. Some bacteria produce chemicals that promote plant growth. Some bacteria help plants resist disease. Some bacteria help plants tolerate environmental stress like drought. And in return, plant roots release chemicals, sugars and other organic compounds that feed the bacteria living near them. It is a quiet partnership, worked out over hundreds of millions of years of shared evolution. Plant and bacterium, root and soil, each one quietly supporting the other in ways neither could fully manage alone. Slowly, gently, let us think about bacteria in the ocean. The ocean covers more than 70% of Earth's surface, and it is teeming with bacteria. In each milliliter of seawater, a space smaller than a sugar cube, there are typically 1 million bacteria. The open ocean is, in terms of bacterial life, one of the most densely populated environments on the planet. Ocean bacteria play an essential role in the marine food web. They decompose organic material. They cycle nutrients. They are themselves eaten by tiny organisms, which are eaten by larger ones, and certain ocean bacteria, called Prochlorophyta and Prochlorococcus, are among the most abundant photosynthetic organisms on Earth. These tiny, quietly extraordinary bacteria are thought to be responsible for producing somewhere between 1/4 and 1/2 of all the oxygen produced on Earth each year. Quietly, at the surface of the sea, billions of tiny bacteria breathe out the air we need. Let us take a quiet moment to think about bacteria that glow. Some bacteria are bioluminescent, meaning they produce their own light through chemical reactions. Bioluminescent bacteria are found primarily in marine environments, in seawater, in decaying organic material, and in the organs of certain deep-sea fish. The light these bacteria produce is a soft, cool blue-green, the result of a chemical reaction involving a protein called luciferase and a molecule called luciferin. This is the same basic chemistry that makes fireflies glow. These bacteria produce light continuously, not in flashes, when present in large numbers. For example, in a bay of warm tropical water stirred by waves at night, they can make the water glow with a soft, ethereal blue-green light. It is one of the most quietly beautiful phenomena in nature. Light made by living things, in the dark of the ocean, for reasons that are still not fully understood. Now, as we continue, let us think about why bioluminescent bacteria produce light at all. This is a question that scientists have been quietly exploring for many years. One idea is that bioluminescence is linked to quorum sensing. The genes that control light production in many bioluminescent bacteria are activated by the same chemical signals that bacteria use to sense population density. So bacteria may only glow when they are in a large enough group, when the quorum signal tells them that there are many others nearby. Some scientists suggest that the glow may attract small organisms that eat the bacteria, and those organisms might then spread the bacteria to new environments. Others suggest that the light might have other functions, or might be a byproduct of other useful chemical reactions. The quiet truth is that we do not yet fully understand it, and there is something gently restful about that. Some things remain softly mysterious, gently moving forward. Let us think about the colors that bacteria produce. Bacteria can produce pigments, colored molecules, in a remarkable variety of hues. The bacterium Chromobacterium violacum produces a deep purple pigment called violation. Serratia marcescens produces a red pigment. Staphylococcus aureus produces a golden yellow pigment. Pseudomonas aeruginosa can produce a striking blue-green color. These pigments serve various biological functions. Some protect bacteria from ultraviolet radiation. Some have antimicrobial properties, helping bacteria compete with other microorganisms in their environment. Some may play roles in signaling, and some, we are honestly not entirely sure. The living world is full of color, and bacteria are no exception. In a petri dish in a laboratory, a bacterial colony can be a remarkably beautiful thing. A soft circle of gold, or pink, or white, or purple, quietly growing. Let us take a quiet moment to think about bacteria that produce silk-like structures. Some bacteria produce something called cellulose, the same material that plant cell walls are made of. Bacterial cellulose is a network of extremely fine, interconnected fibers that form a delicate, gel-like mat around the bacteria producing it. Bacterial cellulose is extraordinarily pure, much purer than plant cellulose, and has remarkable properties. It is strong, flexible, and able to hold a large amount of water. Scientists have explored using bacterial cellulose for a range of applications. As a wound dressing that keeps wounds moist and clean, as a material for transparent screens and sensors, and even as a food product. The bacteria that produce it do so quietly, fiber by fiber, as they grow and divide, building a soft, beautiful network that humans are still learning to appreciate and use. Slowly, gently, let us think about the beautiful and occasionally vivid world of bacteria in hot springs. Certain bacteria called thermophiles form colorful, layered mats around the edges of hot springs, creating the stunning rings of color that make places like Yellowstone National Park so visually striking. The bright yellows, oranges, and greens of these bacterial mats are caused by the pigments the bacteria produce. Different bacteria grow at different temperatures, so as you move from the scalding center of the spring outward to where it is cooler, the community of bacteria changes. Different temperatures, different bacteria, different pigments, different colors. The result is a quietly extraordinary pattern of color, a living painting made by microscopic organisms, growing in rings around a pool of hot water, under an open sky, life arranging itself into beauty without any awareness of what it is creating. Now, as we continue, let us take a quiet moment to think about one of the most important events in the history of life on Earth, and the central role that bacteria played in it. About 2 billion years ago, a profound change began. Certain bacteria, probably ancient ancestors of modern mitochondria, were engulfed by larger cells, but instead of being digested, they survived. They began to live inside the larger cell as a kind of tenant. Over time, the relationship deepened. The engulfed bacteria became more and more integrated into their host. They began to provide the host cell with energy, in exchange for the protection and nutrients the host cell could provide. Eventually, these once independent bacteria became mitochondria, the energy-producing structures found in almost every cell of every animal, plant, and fungus on Earth. The mitochondria in your cells right now are, in the deepest evolutionary sense, ancient bacteria. Let us think gently about what that means. Every time your body uses energy, every heartbeat, every thought, every breath, that energy is produced by structures that began billions of years ago as free-living bacteria. Mitochondria still carry their own small circle of DNA, separate from the DNA in the cell's nucleus. This mitochondrial DNA is a quiet remnant of their independent bacterial past, and it is still inherited differently from the DNA in the nucleus. Mitochondrial DNA is passed almost exclusively through the mother, from mother to child, generation after generation, in an unbroken line going back to that ancient event when a bacterium first took up residence inside another cell. There is something deeply and quietly moving about that. A lineage of mothers, stretching back 2 billion years, quietly passing on what was once a living bacterium. Gently moving forward, let us think about another ancient bacterial structure that has shaped the living world. Stromatolites are layered rock-like structures built by communities of bacteria, primarily cyanobacteria, over extremely long periods of time. As cyanobacteria photosynthesize, they build thin layers of sediment and organic material around themselves. Over thousands of years, these layers accumulate into large, dome-like structures in shallow water. Stromatolites were among the dominant life forms on Earth for billions of years. From about 3.5 billion years ago until the emergence of more complex life began to reduce their prevalence. Today, living stromatolites still exist in a few locations on Earth, most famously in Shark Bay in Western Australia. They are the closest living relatives of the oldest clearly biological structures on the planet. Quiet living monuments to an almost incomprehensibly ancient past. Now, as we continue, let us think about the Great Oxidation Event, one of the most profound and quietly dramatic changes in Earth's history. For the first 2 billion or so years of Earth's existence, the atmosphere contained very little oxygen. The oceans were rich in dissolved iron, and the chemistry of the planet was very different from today. Then, cyanobacteria began to proliferate. Their photosynthesis, which produces oxygen as a byproduct, began to release oxygen into the oceans and atmosphere in large quantities. This oxygen reacted with the dissolved iron in the oceans, forming vast deposits of iron oxide, the rust-red banded iron formations that we mine today as iron ore. Slowly, over hundreds of millions of years, oxygen levels in the atmosphere rose. The planet changed. Many organisms that could not tolerate oxygen went extinct, and new forms of life that could use oxygen to produce energy far more efficiently began to evolve, all because of bacteria. Slowly, gently, let us think about bacteria preserved in amber. Amber is fossilized tree resin, and occasionally it captures organisms that were present millions of years ago. Bacteria have been found preserved in amber that is tens of millions of years old. In some remarkable cases, bacteria have even been found in fluid inclusions inside salt crystals that are hundreds of millions of years old. And in a few extraordinary claims, some scientists have reported reviving bacteria from ancient amber and ancient salt deposits. These claims are controversial and not fully accepted by all scientists. But the possibility, the quiet possibility, that a bacterium might survive sealed within a crystal for 250 million years, and then be gently revived in a modern laboratory, speaks to a kind of durability and persistence that seems almost beyond imagination. Life, waiting softly in stone, for a very long time. Let us take a quiet moment to think about the many ways that bacteria are involved in the foods we eat. Fermentation, the process by which bacteria transform food into other, often more complex and flavorful forms, is one of the oldest and most widely practiced food traditions in the world. Yogurt is made by bacteria. Cheese is made with the help of bacteria. Sourdough bread rises because of bacteria. And yeast, too, but bacteria play a significant role. Sauerkraut, kimchi, pickles, miso, tempeh, kefir, vinegar, natto. All of these foods owe their existence to the quiet work of bacteria. In each case, bacteria consume sugars in the food and produce acids, gases, or other compounds that transform the food's texture, flavor, and preservation. Fermented foods have nourished human populations for thousands of years. And bacteria have been gently doing this work since long before anyone understood how. Now, as we continue, let us think about the bacteria in yogurt. Yogurt is made by adding specific bacteria, most commonly Lactobacillus bulgaricus and Streptococcus thermophilus, to warm milk and allowing them to grow. These bacteria consume the lactose, the sugar, in milk and convert it into lactic acid. As the acid increases, the milk proteins change their structure and the mixture thickens into the creamy, smooth texture we know as yogurt. The whole process takes only a few hours. It is quiet, warm, and gentle, just bacteria, growing slowly in warm milk, producing acid as they go, quietly transforming a liquid into something thick and tangy and nourishing. And when we eat yogurt, we consume those bacteria too, taking living organisms into our digestive system, where they may briefly contribute to the ecology of the gut. Gently moving forward, let us think about sourdough bread and the quiet biology that makes it rise. Sourdough is made using a starter, a mixture of flour and water that has been left to ferment. In that mixture, wild bacteria and yeast from the flour and the environment begin to grow. The bacteria, primarily lactic acid bacteria like Lactobacillus, produce lactic and acetic acids, giving sourdough its characteristic tangy flavor. The yeast produces carbon dioxide gas, which causes the bread to rise, but the bacteria play another role too. They produce enzymes that break down some of the complex molecules in flour, including certain proteins that some people find difficult to digest. The long, slow fermentation of sourdough bread, which can take 12 to 24 hours or more, gives the bacteria plenty of time to do this gentle work. The result is a bread that is, in many ways, more digestible and more nutritious than quickly made bread. Let us take a quiet moment to think about cheese and the remarkable diversity that bacteria make possible. There are hundreds of varieties of cheese in the world, and the specific bacteria involved in each cheese's production, along with the conditions of its aging, largely determine its unique flavor, texture, and aroma. The mild, creamy flavor of a fresh mozzarella comes from a different bacterial community than the sharp, crystalline complexity of a well-aged Parmesan. The blue-green veins running through a Roquefort or Gorgonzola cheese are produced by a mold, but bacteria play important supporting roles in the flavor development. The orange, washed rind of a soft Limburger or Taleggio is produced largely by the surface ripening bacteria growing on it. In each case, bacteria are quietly working, producing acids and enzymes and other compounds, to create something complex and beautiful from something as simple as milk. Slowly, gently, let us think about vinegar. Vinegar has been made by humans for thousands of years, and its production depends on two sequential steps of bacterial fermentation. In the first step, yeast converts the sugars in a fruit juice or grain liquid, apple juice, grape juice, malted barley, into alcohol. Then, in the second step, certain bacteria called acetic acid bacteria convert that alcohol into acetic acid. Acetic acid is what makes vinegar taste sharp and sour. These bacteria, the most well-known of which is Acetobacter, are widespread in nature. They grow on the surface of fermenting liquids, where they can access oxygen. They convert alcohol to acid quietly and efficiently. The result is apple cider vinegar, wine vinegar, balsamic vinegar, rice vinegar, all of them the product of bacteria doing what bacteria have always done, taking one molecule and quietly, gently turning it into another. Now, as we continue, let us think about antibiotics, medicines that have transformed human health, and whose origins lie in the natural behavior of bacteria and fungi. Antibiotics are compounds that inhibit or kill bacteria. Many of them were first discovered as natural products made by bacteria or fungi as a means of competition. The most famous antibiotic, penicillin, was discovered in 1928 when Alexander Fleming noticed that a mold called penicillium was producing a compound that killed the bacteria growing around it. The mold was doing what microorganisms have done for hundreds of millions of years, producing chemicals to defend their space and resources against competitors. Humans learned to recognize this, to extract and purify these compounds, and to use them as medicines. The first antibiotic treatments began in the early 1940s, and they quietly changed the outcome of countless infections that had previously been fatal. Let us think gently about how antibiotics work. Different antibiotics work in different ways, targeting different aspects of bacterial biology. Some antibiotics, like penicillin and its relatives, work by interfering with the construction of the bacterial cell wall. Without a properly formed cell wall, the bacterium cannot maintain its shape, and it eventually bursts. Other antibiotics target the bacterial ribosome, the molecular machine that bacteria use to build proteins, and block protein production. Others target DNA replication. Others disrupt the bacterial cell membrane. The key to an effective antibiotic is that it targets something that is specific to bacteria. Something bacteria have that human cells do not, or something that works differently enough in bacteria that the antibiotic can affect bacteria without harming the patient. This specificity is what makes antibiotics such elegantly quiet tools of medicine. Gently moving forward, let us think about antibiotic resistance. Over time, bacteria can evolve resistance to antibiotics. This happens through the same basic process that drives all evolution: random variation and natural selection. When a population of bacteria is exposed to an antibiotic, most will be killed, but occasionally, a bacterium carries a random mutation that makes it slightly resistant to the antibiotic. That bacterium survives. It reproduces. Its offspring inherit the resistance. Over many generations, resistant bacteria become more common. The more widely and frequently antibiotics are used, the stronger and more consistent the selective pressure for resistance. This is one of the quieter but more significant challenges in modern medicine. Bacteria do not resist antibiotics out of malice or strategy. They simply evolve, as all living things do, in response to the pressures of their environment. Now, as we continue, let us think about bacteria that are genuinely beneficial in medicine, beyond simply being targets for treatment. Bacteria are used to produce many important medical compounds. Insulin, the hormone that people with type 1 diabetes need to survive, was once extracted from the pancreases of pigs and cows. Today, it is produced by bacteria that have been genetically engineered to carry the human insulin gene. The bacteria grow in large fermentation vats, quietly producing human insulin as they go about their normal cellular activities. Many other important proteins used in medicine are similarly produced by bacteria. Growth hormones, clotting factors, vaccines, bacteria, in quiet and controlled environments, serving as tiny living factories for the molecules that keep people healthy. It is a remarkable partnership between microbiology and medicine. Slowly, gently, let us think about probiotics. Probiotics are live bacteria, and sometimes other microorganisms, that are consumed with the intention of benefiting health. The idea behind probiotics is that introducing specific beneficial bacteria into the gut can supplement or restore the natural microbiome, particularly after disruptions like illness or antibiotic treatment. Probiotic products include certain yogurts, fermented milks like kefir, and dietary supplements. The evidence for the health benefits of specific probiotics varies considerably depending on the strain and the condition being targeted. For some conditions, particularly certain types of diarrhea, there is reasonably good evidence that specific probiotics are helpful. For other conditions, the evidence is still being gathered. The science of probiotics is a quiet, ongoing conversation between researchers and the remarkable complexity of the human microbiome. Let us take a quiet moment to think about the living community that exists in soil. A single teaspoon of healthy, fertile soil contains an estimated 1 billion bacteria. Not just one type, but thousands of different species, each playing its own quiet role in the complex ecology of the soil. These bacteria break down organic matter, cycle nutrients, produce acids that weather rocks and release minerals, and interact in complex ways with plant roots, fungi, and other soil organisms. When we walk across a lawn or a field, with every step, we are walking on a surface that is home to an invisible community of extraordinary complexity and richness. The soil beneath our feet is not just dirt. It is a living system, quietly maintained by organisms most of us never think about, performing functions that are essential to everything that grows from the ground. Now, as we continue, let us think more carefully about the bacteria that form partnerships with plant roots. One of the most important of these partnerships is between legume plants. Peas, beans, clover, alfalfa, and bacteria called rhizobia. Rhizobia live in the soil, and when they encounter the roots of a compatible legume, they are attracted toward them and enter the root tissue. The plant forms small, round structures on its roots called nodules, swellings that are visible to the naked eye if you pull a legume plant gently from the ground. Inside these nodules, the rhizobia live in a kind of protected, nutrient-rich environment. In exchange, they fix nitrogen, converting atmospheric nitrogen gas into ammonia that the plant can absorb. The plant feeds the bacteria. The bacteria nourish the plant. A quiet, ancient partnership, running continuously in millions of fields and meadows. Gently moving forward, there is a genus of bacteria called actinomyces, or more broadly actinobacteria, that lives abundantly in soil and plays a particularly important role in decomposition. Actinobacteria produce a network of thread-like filaments, similar in some ways to the structure of fungi. Though bacteria and fungi are entirely unrelated, these filaments help break down tough plant material like cellulose and lignin, the complex structural compounds that make up wood and the cell walls of plants. Actinobacteria are also responsible for a distinctive smell that many people find deeply pleasant and comforting: the earthy smell of soil after rain. This smell is produced by a compound called geosmin, which actinobacteria release as they go about their quiet work. The smell of rain on dry earth, that soft, ancient, peaceful scent, is the smell of bacteria at work in the soil. Let us take a quiet moment to think about bacteria and the slow, patient process of soil formation. Soil does not form quickly. It forms over thousands and millions of years, through the combined action of physical weathering, chemical reactions, and biological processes. And bacteria are among the earliest pioneers of soil formation. On a newly exposed rock surface, after a glacier retreats, or a volcanic eruption deposits fresh lava, bacteria are often among the first organisms to arrive and establish themselves. They colonize the bare rock, producing acids that slowly dissolve and break down the rock surface. As bacteria live and die on the rock, they contribute organic matter, over long periods of time. This thin biological layer begins to create the conditions in which more complex soil communities can develop. The very soil that feeds the world began, in many places, with bacteria quietly colonizing bare stone. Slowly, gently, let us think about what happens to bacteria when the soil becomes very dry. Some bacteria in soil form spores when conditions become unfavorable, entering a state of dormancy until moisture returns. Others have thick, waxy cell membranes or produce protective molecules that help them retain water and survive desiccation. When rain finally comes to dry soil, and this is connected to what we were saying earlier about geosmin, the bacteria that have been dormant in the soil begin to revive and become active again. They start metabolizing, reproducing, releasing geosmin and other molecules into the air. This is part of what we smell when rain falls on dry earth. The first drops of rain on dry soil release a burst of scent that includes geosmin from the reviving bacteria. It is the smell of life quietly waking up after rest. Now, as we continue, let us think about bacteria that live in conditions of extreme temperature. The bacteria most adapted to heat are called hyperthermophiles, and some of them can survive and grow at temperatures that exceed 100 degrees Celsius, the normal boiling point of water. They live near deep-sea hydrothermal vents, where water superheated by volcanic activity is forced out under such high pressure that it remains liquid even at these extreme temperatures. These bacteria have evolved proteins that remain stable and functional at temperatures that would cause most proteins to unravel and stop working. Their enzymes, including DNA polymerases, the enzymes that copy DNA, have become extraordinarily useful in biotechnology. One enzyme from a thermophilic bacterium, Taq polymerase, is the key ingredient in a widely used laboratory technique called PCR that allows scientists to make millions of copies of specific DNA sequences. Hot spring bacteria, quietly enabling modern genetics. Let us think gently about bacteria that live in ice. Psychrophilic bacteria, cold-loving bacteria, have evolved mechanisms to function at temperatures near and even below 0 degrees Celsius, in the liquid water trapped within the crystal structure of ice, and in the extremely cold waters of polar oceans. These bacteria carry out their slow, patient lives. Their cell membranes are adapted to remain flexible at low temperatures. They contain higher proportions of unsaturated fatty acids that do not solidify in the cold the way saturated ones do. Their enzymes are adapted to function at low temperatures, where the biochemical reactions of life proceed far more slowly. Psychrophilic bacteria play an important role in cold ecosystems. They are among the primary decomposers in polar regions, and they contribute to nutrient cycling in ocean waters that are too cold for most other organisms to function effectively in. Gently moving forward, let us think about bacteria that live in highly acidic environments. Acid mine drainage is one of the most extreme acid environments on Earth. When sulfide minerals in mines are exposed to air and water, certain bacteria, called acidophiles, catalyze the oxidation of those minerals, producing sulfuric acid. The water that drains from some mines is extraordinarily acidic, sometimes reaching a pH close to 0, and yet bacteria thrive in it. Aciditheobacillus ferrooxidans is one such bacterium, able to grow at pH values below 1. These bacteria have specialized proton pumps that actively keep their internal environment at a near-neutral pH, even while the external environment is intensely acidic. Their outer membranes are adapted to resist the corrosive effects of the acid. They thrive in a chemical environment that would dissolve most materials. Quietly, persistently, doing what bacteria have always done. Now, as we continue, Let us think about bacteria that survive in environments of extremely high salt concentration. Halophilic bacteria, salt-loving bacteria, are found in salt lakes, salt flats, and highly saline coastal environments. The Dead Sea, one of the saltiest bodies of water on Earth, with salinity about 10 times that of the ocean, is home to halophilic bacteria. These bacteria have evolved to require high salt concentrations to maintain their structure. Most bacteria would shrivel and die in such conditions, because water would be drawn out of their cells by osmosis. Halophilic bacteria counteract this by accumulating high concentrations of compatible solutes, molecules that balance the osmotic pressure without interfering with the bacteria's biochemistry. In some halophilic bacteria, even the internal machinery of the cell is adapted to function best in the presence of very high salt concentrations. Life, adjusted to conditions that seem impossible. Slowly, gently, let us think about bacteria that live deep in the Earth's crust. This is perhaps one of the most quietly astonishing environments that bacteria have colonized. Several kilometers below the Earth's surface, in rock fractures, and in the water trapped within those fractures, bacteria have been found living in conditions of complete darkness, extreme pressure, very little oxygen, and extremely slow nutrient supply. These deep subsurface bacteria may have metabolisms that are almost incomprehensibly slow by surface standards. Some researchers have suggested that certain deep subsurface bacteria may have generation times, the time between one cell division and the next, measured not in minutes or hours, but in thousands of years. Essentially, these bacteria are nearly stationary in time, very slowly dividing, very slowly changing, living out a kind of existence that is as different from the rapid, active life of a surface bacterium as we can imagine, quiet beyond all ordinary quiet. Let us take a quiet moment to think about how some bacteria respond to light. Like plants, some bacteria have evolved the ability to sense light and use it to their advantage. Cyanobacteria and other photosynthetic bacteria can orient themselves toward light, a process called phototaxis. They move toward wavelengths of light that are most useful for photosynthesis, and away from wavelengths or intensities that might be harmful. This movement is guided by light-sensitive proteins in the bacterial cell membrane. Some bacteria also use light in a very different way, not for energy, but for sensing time. Cyanobacteria have what may be the simplest known circadian clock, an internal timekeeping system that cycles with a roughly 24-hour period, even in the absence of external light cues. This quiet internal rhythm allows the bacterium to anticipate the coming of day and night, and to time its activities accordingly. Now, as we continue, let us think about a remarkable and recently discovered form of bacterial photosynthesis. Most photosynthetic bacteria that we are familiar with use visible light, the same spectrum of light that plants use. But some bacteria can use infrared light, light with wavelengths beyond what the human eye can detect. In the heat-generating range of the spectrum, these bacteria use a pigment called bacteria chlorophyll, which absorbs infrared light. Some of them can even photosynthesize using light that is so far into the near-infrared range that it was previously thought to carry insufficient energy to drive the chemistry of photosynthesis. The discovery of these far-red and near-infrared photosynthesizing bacteria expanded our understanding of what is possible in biology. If bacteria can harvest energy from wavelengths of light we previously thought were too weak, then perhaps life in other places, on other worlds, orbiting cooler, dimmer suns, might photosynthesize in ways we have not yet imagined. Gently moving forward, let us think about how light affects the colors of bacteria that we see. Many bacteria produce pigments whose primary function is photoprotection, shielding the bacterium from the potentially damaging effects of ultraviolet radiation and high-intensity visible light. Carotenoid pigments, for example, which give some bacteria their yellow, orange, and red colors, can absorb and dissipate excess light energy, protecting the cell's machinery from radiation damage. The same class of pigments, carotenoids, gives carrots their orange color, and autumn leaves their yellow and orange tones. So when you see the soft orange of a ripe carrot, or the gold of a turning leaf in autumn, you are seeing the same family of protective pigment that certain bacteria use to shield themselves from the sun. A quiet connection between the microscopic and the everyday. Let us take a quiet moment to think about the role of bacteria in producing the clear, blue color of some glacial lakes. Glacial meltwater carries rock flour, extremely fine particles of rock ground down by the movement of ice. When this fine sediment enters a lake, it remains suspended in the water for a long time, giving the water a milky, turquoise appearance. Bacteria play a role in eventually clearing this suspended sediment, as they colonize and break down the organic matter that slowly settles with it. But even beyond this, bacterial communities in glacial lakes are thought to contribute to the cycling of minerals and nutrients in these cold, low-nutrient waters. In some glacial lakes, bacterial biofilms on the lake floor are among the dominant forms of life, quietly processing the little energy available to them in these cold, clear, remarkably still environments. Slowly, gently, let us think about how darkness affects bacterial communities. In environments without light, in deep caves, in deep ocean sediments, in deep groundwater, bacteria that depend on sunlight cannot survive. Instead, the bacteria found in these dark environments derive their energy from chemical reactions involving inorganic molecules. These communities are called chemolithotrophic, rock chemical feeding, in the absence of the sun, which drives almost all surface life either directly through photosynthesis or indirectly through the food chains it supports. These deep, dark bacterial communities are entirely self-sustaining. They do not depend on sunlight. They do not depend on photosynthesis. They extract energy entirely from the chemistry of the rocks and water around them. And we can take a moment to notice how profoundly independent that is. Life, finding its own quiet energy, in the total absence of light. Now, as we continue, let us think about the remarkable bacteria that live in the digestive systems of cattle, sheep, and other ruminants. Ruminants have a specialized stomach compartment called the rumen, which acts as a fermentation chamber. In this warm, moist, oxygen-free environment, a complex community of bacteria and other microorganisms breaks down the cellulose in grass and other plant material. Cellulose is the primary structural component of plant cell walls, and it is one of the most abundant organic molecules on Earth. Most animals cannot digest it at all, but the bacteria in a ruminant's rumen produce enzymes called cellulases that break cellulose down into simpler sugars, which the bacteria then ferment. The products of this fermentation, including volatile fatty acids, are absorbed by the ruminant and used as an energy source. Without these bacteria, a cow could not digest grass. The peaceful grazing of cattle in a field depends entirely on microscopic bacterial work. Let us think gently about bacteria in the digestive systems of insects. Many insects depend on bacteria to help them digest their food or to supplement their nutrition. Termites, for example, despite their small size, are capable of consuming and digesting wood, one of the toughest organic materials on Earth. They can do this not alone, but with the help of microorganisms in their gut, including bacteria, that produce the enzymes needed to break down cellulose and lignin. Without these gut microorganisms, termites cannot digest wood. Similarly, many aphids, small sap-sucking insects, depend on bacterial endosymbionts that live within specialized cells inside the aphid's body. These bacteria, called Buchnera, produce essential amino acids that the aphid cannot make itself, and that plant sap does not provide in sufficient quantities. Without Buchnera, the aphid would be nutritionally deficient. With it, the aphid quietly thrives on a diet that would otherwise be inadequate. Gently moving forward, let us think about the relationship between certain deep-sea animals and chemosynthetic bacteria around deep-sea hydrothermal vents, where hot, chemical-rich water flows from the ocean floor. There are thriving communities of animals, tube worms, clams, mussels, and others. In the total absence of sunlight, these animals derive their nutrition, directly or indirectly, from bacteria that use the chemical energy in the vent water to produce organic molecules through chemosynthesis rather than photosynthesis. Giant tube worms, for example, can grow over a meter long and live clustered around vents. They have no mouth and no digestive system. Instead, they have a specialized organ called a trophosome, packed with chemosynthetic bacteria. The bacteria live inside the worm, producing nutrients from the vent chemicals. The worm provides the bacteria with access to the chemicals they need. Together, in the complete darkness of the deep ocean floor, they sustain each other. Now, as we continue, let us think about bacteria that live in symbiosis with certain marine invertebrates. The Hawaiian bobtail squid has a remarkable relationship with a bioluminescent bacterium called Vibrio fischeri. This tiny squid is active at night, and it would be vulnerable to predators looking up from below, because its dark silhouette against the faint moonlight filtering down from the surface would give it away. But the squid has a specialized light organ in its mantle that is home to a dense colony of Vibrio fischeri. These bioluminescent bacteria glow continuously within the light organ. The squid uses this bacterial glow to match the faint light coming from above, effectively eliminating its own shadow. This phenomenon is called counterlumination. The squid controls the brightness of the bacterial glow using a system of filters and mirrors in the light organ. Bacteria, quietly glowing inside a squid, helping it hide in the dark ocean. Slowly, gently, let us think about the bacteria that live in the gut of honeybees. Bees, like many animals, have a microbiome, a community of bacteria living in their digestive tract. The bee gut microbiome is actually quite simple compared to mammalian gut microbiomes. It consists of only 8 to 10 core bacterial species. But these are remarkably consistent across honeybee species and populations around the world. These core bacteria are thought to be horizontally transmitted between bees through social contact, through the sharing of food, through the close contact of life in a hive. They help protect bees against pathogens, help digest certain components of their diet, and may play a role in immune function. When bee populations come under stress, from pesticides, from habitat loss, from disease, the stability of this gentle bacterial community can be disrupted. And when that happens, the health of individual bees, and potentially the colony, can suffer. Let us take a quiet moment to think about the role of bacteria in the water cycle. It might not seem obvious at first that bacteria have anything to do with rain, but they do. Clouds form when water vapor condenses around tiny particles in the atmosphere, called cloud condensation nuclei. Dust, sea salt, and other particles can serve as these nuclei. And so can bacteria. Certain bacteria, particularly Pseudomonas syringae, are extremely efficient cloud condensation nuclei. They produce proteins on their outer surface that can initiate ice crystal formation at temperatures where ice would not normally form. These bacteria are found in the atmosphere, having been lofted up from the surfaces of plants and soil by wind. When they encounter water vapor in clouds, they can trigger the formation of ice crystals, and ultimately precipitation. Rain, falling softly from clouds, may in some cases be carrying the bacteria that helped form it. Now, as we continue, let us think about the bacteria floating in the air around us right now. The atmosphere contains a significant and diverse community of airborne bacteria. These bacteria are not actively growing or dividing in the air. They are simply being transported, suspended in droplets of water or attached to dust particles. They come from soil surfaces, from the ocean, from the surfaces of plants, from animal activities. Studies of the air in different environments have found hundreds of different bacterial species present even in clean outdoor air. Most of these bacteria are harmless. They are simply passing through, carried by wind currents, some of them traveling extraordinary distances. Bacteria have been collected from the atmosphere at altitudes of many kilometers. Some may travel across oceans, carried by upper-level air currents, before eventually settling back to Earth. Quiet travelers in the invisible river of air, gently moving forward. Let us think about bacteria and the production of greenhouse gases. Bacteria play important roles in the production and consumption of several gases that affect Earth's climate. Methane, for example, is produced by a group of archaea, which are related to but distinct from bacteria, called methanogens. But bacteria also produce and consume methane in various environments. More directly relevant are the bacteria involved in the nitrogen cycle. Certain bacteria produce nitrous oxide, a potent greenhouse gas, as a byproduct of the process of denitrification, by which they convert nitrate back into nitrogen gas. Nitrous oxide is about 270 times more potent as a greenhouse gas than carbon dioxide over a 100-year period. The bacteria producing it are not doing anything harmful from their own perspective. They are simply performing their ancient metabolic work. But that work intersects, quietly, with the changing chemistry of the atmosphere we all share. Let us take a quiet moment to think about dimethylsulfonopropionate, or DMSP. This is a compound produced in enormous quantities by marine algae and certain bacteria in the ocean. When these organisms are eaten or die, DMSP is released and broken down by marine bacteria. One of the products of this breakdown is dimethyl sulfide, or DMS. DMS evaporates from the ocean surface into the atmosphere, where it oxidizes to form sulfate particles. These particles can act as cloud condensation nuclei, contributing to the formation of clouds over the ocean. So certain marine bacteria, by consuming DMSB and releasing DMS, may influence cloud formation over the sea, which in turn influences how much sunlight reaches the ocean surface, which in turn influences the growth of the organisms that produce the DMSB in the first place. A quiet, circular feedback loop, connecting bacteria to clouds to sunlight to life. Slowly, gently, let us think about the way bacteria help cleanse the atmosphere of certain pollutants. Some bacteria are capable of breaking down organic pollutants, volatile organic compounds, certain hydrocarbons that would otherwise accumulate in the atmosphere or in soil and water. The ability of bacteria to break down complex organic molecules means that they can sometimes metabolize pollutants that enter their environment. This property of bacteria, bioremediation, is used intentionally in some cases to help clean up contaminated sites. Bacteria have been used to help break down oil spills in the ocean, to degrade pesticides in contaminated soil, and to process industrial waste in treatment facilities. The same quiet metabolic flexibility that allows bacteria to colonize every environment on Earth also equips them to slowly, gently, process some of the chemical messes that human activity has created. Life, quietly cleaning up after itself and after others. Now, as we continue, let us think about how bacteria became central to the science of genetics. In the mid-20th century, much of what we know today about how genes work was first discovered in bacteria. Bacteria are ideal for genetic research in many ways. They are simple. They reproduce quickly. They can be grown in enormous numbers in small spaces. And they have relatively small genomes that were, at the time, more tractable to study than the genomes of larger organisms. The discovery that DNA is the carrier of genetic information. The famous Avery MacLeod-McCarty experiment in 1944 used bacteria. The discovery of the double helix structure of DNA by Watson and Crick was supported by X-ray crystallography data. But much of the work that confirmed how DNA is read and copied was done in bacteria. Much of what we know about transcription, translation, and gene regulation was worked out in the bacterium Escherichia coli. Quietly, bacteria taught us most of what we know about the fundamental machinery of life. Let us think gently about CRISPR, one of the most significant scientific developments in recent years, and its bacterial origins. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is a system that bacteria evolved as a form of immune memory against viruses. When a bacterium survives a viral attack, it can incorporate a small fragment of the virus's DNA into a special region of its own genome, the CRISPR region. If the same virus attacks again, the bacterium can recognize it using the stored fragment and mount a targeted response, using proteins called Cas to cut the viral DNA and disable it. Scientists discovered this system and developed a way to use it as a precise tool for editing DNA in any organism. Today, CRISPR is used in research on genetic diseases, in the development of new medicines, and in agricultural biotechnology. A bacterial immune system evolved over billions of years for a very different purpose, quietly becoming one of the most powerful tools in modern biology. Gently moving forward, let us think about the coli bacterium, which is one of the most studied organisms on Earth. Escherichia coli, named for the German bacteriologist Theodor Escherichia, who first described it in 1886, is a rod-shaped bacterium that lives in the intestines of mammals, including humans. It is normally harmless and is, in fact, a normal and beneficial member of the gut microbiome. But E. coli has also become the workhorse of laboratory biology. It grows quickly. It is easy to manipulate genetically. Its biology is so well understood that it has been used to produce countless research products, including insulin and other pharmaceuticals, and to test and develop biotechnology techniques. The complete genome sequence of E. coli was published in 1997, a milestone in genomics. This quiet, rod-shaped inhabitant of our intestines has, through science, become one of the most significant organisms in the history of human health. Now, as we continue, let us think about the discovery of penicillin in a little more detail, because it is one of those gentle stories in science that is worth sitting with quietly. In September 1928, Alexander Fleming returned to his laboratory at St. Mary's Hospital in London after a holiday. on one of his petri dishes, which had been left exposed. He noticed something unusual. A mold had contaminated the plate. Around the mold, there was a clear zone, an area where the bacteria that had been growing on the plate were absent. The mold was preventing bacteria from growing near it. Fleming was curious. He identified the mold as Penicillium notatum and began to investigate the substance it was producing. He named it penicillin. It took more than a decade for others, Howard Florey and Ernst Chain, to develop penicillin into a medicine that could be used in patients. But it began with one quiet observation. One scientist noticing something unusual on a contaminated petri dish. Slowly, gently, let us think about what bacteria have taught us about the origin of life. One of the deepest and most fundamental questions in science is how life began on Earth. And bacteria, particularly the simplest and most ancient-seeming bacteria, offer some of the best clues we have. The deep branching groups of bacteria and archaea that live in extreme environments. Hydrothermal vents, hot springs, acidic pools, are thought to resemble, at least in some ways, the kinds of organisms that may have existed very early in Earth's history. By studying their biochemistry, their membranes, their energy-generating mechanisms, and their genetics, scientists have gradually built up a picture of what the earliest life might have looked like. Not photosynthetic, not feeding on complex organic molecules, perhaps living on hydrogen and carbon dioxide near hot springs, quietly extracting chemical energy from the geology of the early Earth. Let us take a quiet moment to think about bacteria and the future of medicine. There is a field called phage therapy that is attracting renewed interest. As antibiotic resistance becomes more challenging, bacteriophages, or simply phages, are viruses that infect bacteria. They are extraordinarily specific. A phage that infects one type of bacterium will generally not infect others. The idea of using phages to treat bacterial infections is not new. It was explored in the 1920s and 1930s, but the development of antibiotics largely overshadowed it. Now, with antibiotic-resistant bacteria becoming more common, phage therapy is being reconsidered. Because phages evolve along with bacteria, they may be able to keep pace with evolving resistance in ways that fixed antibiotic molecules cannot. The use of viruses that target specific bacteria to treat infections that antibiotics can no longer reach is a quiet and elegant idea, drawn from the ancient biological arms race between bacteria and the viruses that prey on them. Now, as we continue, let us think about bacteria and the future of materials science. Bacteria produce a remarkable variety of materials: bioplastics, cellulose, silk-like fibers, minerals, and scientists are exploring how to harness these biological production systems for useful purposes. Some bacteria produce polyhydroxyalkanoates, or PHAs, biodegradable plastics, that they accumulate inside their cells as energy storage compounds. Unlike conventional plastics, PHAs break down naturally in the environment. Several companies are now using bacteria to produce PHAs at industrial scale as a more environmentally friendly alternative to petroleum-based plastics. Other researchers are using bacteria to produce biosensors, electronic components, and even to assist in the construction of nanoscale structures. The metabolic versatility of bacteria, their ability to produce an enormous diversity of molecules, makes them potentially powerful allies in the development of more sustainable materials and technologies. Gently moving forward, let us think about bacteria and space exploration. Astrobiologists, scientists who study the possibility of life beyond Earth, study extremophilic bacteria on Earth. as models for what life might look like in other environments in our solar system. The discovery that bacteria can thrive in the cold, salty water beneath Antarctic ice sheets, in conditions similar to what might exist in the subsurface oceans of Europa or Enceladus, moons of Jupiter and Saturn respectively, has expanded our sense of where life might be found elsewhere. Bacteria that survive high radiation environments are studied as models for potential life on Mars, where the thin atmosphere provides little shielding from cosmic radiation, and bacteria have been sent to space. Some have been found to survive on the outside of the International Space Station for months at a time, not on Earth, not in the controlled warmth of a laboratory, but in the vacuum and radiation of outer space, quietly persistent. Even there, let us take a quiet moment to think about bacteria and renewable energy. Certain bacteria can be used to produce biofuels. Some bacteria produce hydrogen gas as a byproduct of their metabolism. Others can be used to produce ethanol or other liquid fuels. From plant waste, microbial fuel cells use the metabolic activity of bacteria to generate electricity directly from organic matter. In a microbial fuel cell, bacteria oxidize organic compounds, and in doing so release electrons, which are captured as electrical current. These systems can be used to generate small amounts of electricity while simultaneously treating wastewater. The idea of bacteria quietly producing clean energy from waste, taking material that would otherwise be discarded and converting it into electricity, is one of those quietly beautiful possibilities that sits at the intersection of biology and engineering. Life, as it has always done, finding a way to extract energy from whatever is available. Slowly, gently, let us think about the possibility that bacteria will outlast us. Bacteria have already survived 5 mass extinction events in Earth's history. The asteroid impact that ended the age of the dinosaurs 66 million years ago, catastrophic as it was for large animals, barely touched the bacterial world. Bacteria survived because of their small size, their metabolic diversity, their ability to form spores, their presence in every conceivable environment, and their ability to adapt rapidly to changing conditions. If the human species were to disappear tomorrow, if some disaster were to end our chapter of Earth's story, bacteria would continue. They would continue in the deep rocks. They would continue in the cold ocean. They would continue in the soil and on the surfaces of whatever remained. They were here before us, and in all probability, they will be here long, long after, quietly, persistently, doing what they have always done. Now, as we continue into this later, quieter part of our time together, let us think about the beauty of bacterial colonies growing in a petri dish. When bacteria are grown on a solid medium, in a laboratory, they form colonies, circular patches of growth. Each colony descended from a single original cell. The shape and appearance of these colonies can be remarkably varied and beautiful. Some colonies have smooth, round edges. Some have irregular, feathered edges that look like tiny clouds or snowflakes. Some grow in concentric rings. Some produce pigment, and so appear in vivid colors: white, cream, yellow, orange, pink, red, purple. A plate of mixed bacterial colonies can look like a tiny, abstract painting. Scientists use the appearance of colonies as one way to identify bacteria, but beyond the science, there is just beauty there. The quiet, accidental beauty of tiny living things growing in their own particular ways. Let us think gently about the symmetry that appears in certain bacterial structures. Many bacterial proteins and molecular machines have beautiful, precise, geometric symmetry. At the molecular scale, the flagellar basal body, which we thought about earlier, has a ring structure with rotational symmetry. Many bacterial pili are helical structures with precise repeating geometry. Some bacterial toxins form perfectly symmetric ring structures. When they bind together, and bacterial phages, the viruses that infect bacteria, often have icosahedral heads, shaped like 20-sided polygons, with beautiful, exact geometric symmetry. These symmetrical structures arise not through any deliberate design, but through the physics and chemistry of protein folding and self-assembly. Proteins with complementary shapes snap together spontaneously, and the most stable arrangement is often the most symmetrical one. Nature, and particularly the small biological world, is full of this kind of quiet, effortless, geometric beauty. Gently moving forward, let us think about the diversity of bacterial morphology beyond the basic shapes we considered earlier. While most bacteria are either spheres, rods, or spirals, there are notable exceptions. Stellobacteria have a star shape with flat, thin projections radiating outward. Prosthecobacter bacteria have long, thin extensions called prosthecae, giving them an almost tentacle-like appearance. Some bacteria form flattened, square or rectangular cells, almost crystalline-looking, like the Archean haloquadratum Walsbii, which lives in salt flats, and whose perfectly square, flat shape is unlike almost anything else in the living world. Some bacteria form complex branching networks. Others, like Streptomyces, form intricate fungus-like mycelia. The basic blueprint of a bacterium, a simple cell with a membrane and some DNA, turns out to be capable of an extraordinary variety of expression, quiet creativity, in form, across billions of years. Now, as we continue, let us think about the movements of bacteria under a microscope, because watching living bacteria move is one of the most quietly hypnotic experiences available to anyone with a microscope and the patience to look. Free-swimming bacteria dart and pause and change direction. They trace curved paths through the liquid, adjusting their trajectory in smooth, continuous curves. When they change direction, they tumble, briefly spinning, without any particular orientation, before resuming a new direction of movement. This motion is called a run-and-tumble movement, and it is the characteristic way in which many flagellated bacteria navigate through liquid, just to lie still and watch this, to see these tiny living things moving through their microscopic world, running and tumbling, occasionally pausing, changing direction, is to be quietly reminded that life at every scale has its own kind of motion, its own kind of rhythm, its own kind of quiet purpose. Slowly, gently, we have now arrived at our 100th fact together, and it seems right to take a moment here to reflect quietly on what we have been exploring. We have thought about bacteria that are older than the oceans as we know them. We have thought about bacteria that glow in the dark, that eat rocks, that breathe in ways we cannot, that live in fire and ice and salt and deep stone. We have thought about bacteria inside our own bodies, in our guts, and on our skin, quietly doing things that keep us healthy in ways we rarely think about. We have thought about bacteria in the food we eat, in the soil beneath our feet, in the air we breathe, in the clouds above us, and across all of it, there is a kind of quiet constancy. Bacteria do not hurry. They do not worry. They simply continue. There is something deeply restful about that. Now, as we gently continue toward the end of our journey, let us think about something called horizontal gene transfer, and how it shapes the evolution of bacteria. In most organisms we are familiar with, plants, animals, fungi, genes are passed vertically, from parent to offspring. Genetic variation accumulates slowly, over many generations, but bacteria can also transfer genes horizontally, between individuals that are not in a parent-offspring relationship, even between individuals of different species. We have already talked about conjugation and transformation. There is also a third mechanism called transduction, in which bacteriophages, viruses that infect bacteria, carry pieces of bacterial DNA from one cell to another. Through these mechanisms, bacteria can acquire new genetic capabilities remarkably quickly, not just over many generations, but within a single lifetime. This ability to exchange genetic information horizontally makes bacteria extraordinarily adaptable. A useful gene in one species can spread quietly and efficiently across a wide community. Let us think gently about magnetotactic bacteria, bacteria that can sense the Earth's magnetic field. These bacteria contain chains of tiny magnetic crystals made of magnetite or graphite inside their cells. These crystals, called magnetosomes, are arranged in a row along the length of the bacterium, forming a tiny internal compass needle. The bacteria use this internal magnet to orient themselves along the Earth's magnetic field lines. This orientation seems to help them navigate toward the sediment-water interface. In aquatic environments, where the oxygen levels they prefer are typically found, magnetotactic bacteria have been found in freshwater, marine, and estuarine sediments around the world, and we can take a moment to notice something quietly wonderful about them. Tiny living things, carrying within their own bodies a compass made of crystal, navigating by the magnetic field of the whole Earth. Something so small, orienting itself by something so large, gently moving forward. Let us think about bacteria that produce antibiotics naturally, and the ecology of this chemical warfare. In the soil, bacteria and fungi compete intensely for nutrients and space. Over millions of years, certain bacteria have evolved the ability to produce chemicals that inhibit or kill competing bacteria. These natural antibiotics are part of the ongoing chemical competition between microorganisms in the soil. The actinobacteria we mentioned earlier, the ones responsible for the earthy smell of soil, are particularly prolific producers of antibiotic compounds. Most of the antibiotics used in medicine today were originally discovered as natural products from soil bacteria, particularly from the genus Streptomyces. Streptomycin, tetracycline erythromycin, and many others all came originally from the quiet chemical warfare being waged in the soil beneath our feet. We borrowed these tools from bacteria. We did not invent them. We found them. Now, as we continue, let us think about bacteria and light once more, in a different and softer way. Some bacteria can actually sense the polarization of light. Light from the sky is polarized by its passage through the atmosphere. The angle of polarization varies, in a predictable way, around the position of the sun. Some animals, bees for example, use polarized light as a navigational cue, and there is evidence that some photosynthetic bacteria may also be able to detect light polarization, potentially using it for orientation. The ability of any organism to sense something as subtle as the polarization angle of light is in itself one of those quietly remarkable things that biology occasionally produces. The world is full of information that we humans simply cannot perceive with our unaided senses, and bacteria, in their quiet way, can access at least some of that information and use it to navigate their world. Slowly, gently, Let us think about the way some bacteria produce spore-like structures called cysts. Cysts are different from the endospores we thought about earlier. They are formed by some bacteria as a response to environmental stress, but they are typically not as dramatically durable as true endospores. A cyst is essentially a resting cell. The bacterium rounds up, modifies its cell wall, reduces its metabolic activity, and waits. When conditions improve, the cyst germinates and resumes active life. Some bacteria that form cysts, like Azotobacter, a nitrogen-fixing soil bacterium, can survive for many years in the cyst form, even in dry soil. Cyst formation is one of the many quiet strategies that bacteria have developed for persisting through difficult times, not fighting the difficulty, not trying to overcome it, simply waiting in a state of quiet patience until the conditions are right to resume. Let us take a quiet moment to think about a remarkable kind of bacteria called myxobacteria. These bacteria are unusual in many ways. They are large by bacterial standards. They are able to glide across surfaces, and they exhibit a remarkable form of social behavior. Myxobacteria typically live in groups, moving together across a surface and collectively hunting for prey. When they encounter other bacteria or bacterial colonies, they release enzymes and antibiotics that kill the prey and then absorb the released nutrients. This cooperative predation is sophisticated enough that some scientists describe myxobacteria as having primitive multicellular behavior. And when conditions become difficult and food becomes scarce, myxobacteria do something truly extraordinary. They come together in enormous numbers and build a complex multicellular fruiting body, a stalk structure containing hundreds of thousands of cells. Some cells differentiate to form the stalk, others form myxospores. At the top, a simple bacterium, building something that resembles, in its quiet way, a miniature mushroom. Now, as we continue, let us think about the way bacteria measure time. We mentioned earlier that cyanobacteria have a circadian clock, but the precision and simplicity of this clock are worth thinking about more carefully. The cyanobacterial circadian clock involves just 3 proteins, Ca, Cabe, and Casein. These 3 proteins interact with each other in a cycle that takes approximately 24 hours to complete. Casein can be phosphorylated, have a phosphate group added to it, and dephosphorylated, have the group removed, in a cycle driven by the other 2 proteins. This cycle continues, even in a test tube, outside of any living cell, in a solution containing only the 3 proteins, an ADP, a clock that ticks in a simple solution of 3 proteins, no membranes, no cells, no complex machinery, just 3 molecules, cycling through their interactions, keeping time, the simplest biological clock we know of, ticking quietly away, gently moving forward. Let us think about how bacteria can sense touch. Some bacteria are able to respond to physical contact with surfaces, a phenomenon called mechanosensing. When a swimming bacterium first encounters a surface and contacts it, it may receive a mechanical signal that triggers changes in its behavior, causing it to slow, to increase its attachment to the surface, and to begin the process of forming a biofilm. Some bacteria have surface structures that are thought to act as mechanosensors, responding to the physical force of contact with a solid surface. This is quite extraordinary. When you think about it, a single cell with no nervous system, no brain, no muscles, can sense. When it touches a surface, it can feel, in a purely mechanical chemical sense, the difference between swimming freely in liquid and being in contact with something solid, and it responds to that feeling by beginning a new kind of life, Now, as we continue, slowly and gently, let us think about the role of bacteria in the geologic record. Bacteria have left traces of their presence in rocks dating back billions of years. Stromatolites, as we discussed, are perhaps the most visible, but there are also microscopic traces, chemical signatures called biomarkers, patterns of isotopic ratios in carbon and sulfur that only biological processes can produce, and tiny structures in ancient rocks that may be fossilized cells or cell contents. Studying these ancient bacterial traces allows scientists to reconstruct the history of life on Earth, to understand when different metabolic strategies evolved, when oxygen first appeared in significant quantities, when different groups of bacteria diversified. The bacterial fossil record is not rich in the way that the fossil records of large animals are, but it is extraordinarily ancient, and it speaks quietly across billions of years to those patient enough to listen. Slowly, gently, let us think about something called the last universal common ancestor, or LUCA. This is the hypothetical organism from which all life on Earth is believed to have descended. LUCA was not the first living thing. Life presumably evolved through simpler stages before reaching LUCA, but LUCA is the most recent ancestor, shared by all three domains of life: bacteria, archaea, and eukaryotes. Scientists try to reconstruct what LUCA might have been like by comparing genes that are shared across all three domains. The picture that emerges is of an organism living in a hot, chemically rich environment, possibly a hydrothermal vent that was anaerobic, that used hydrogen and carbon dioxide for energy, and that had a sophisticated genetic system, including ribosomes, and a genetic code similar to our own. LUCA was almost certainly something like a bacterium, not any bacterium alive today, but something small, simple, and quietly ancient, from which all of us, every living thing on Earth, ultimately descended. Let us take a quiet moment now, as we near the end of our journey together, to think about the bacteria that live in the human mouth. The oral microbiome is one of the most diverse and well-studied microbial communities. In the human body, it contains somewhere between 500 and 700 different species of bacteria, living on the teeth, the gums, the tongue, the palate, and the inner surfaces of the cheeks. These bacteria form complex, multi-species biofilms. Dental plaque is one of these biofilms. Most oral bacteria are harmless or beneficial, helping to protect against harmful microorganisms, playing roles in the chemistry of saliva, and participating in processes that help maintain the health of the oral tissue. The oral microbiome is established early in life and tends to be relatively stable over time. It is influenced by diet, by hygiene practices, by the health of the teeth and gums, and by many other factors. A quiet community, living in the warmth of the mouth, doing what bacterial communities do. Now, as we continue, gently, let us think about the bacteria that are involved in the formation of kidney stones. This is a gentle example of bacteria being involved in something that might seem entirely non-biological. Kidney stones are deposits of calcium and other minerals that can form in the kidneys. Certain bacteria, particularly Proteus mirabilis, produce an enzyme called urease, which breaks down urea in urine to produce ammonia. This raises the pH of the urine, makes it more alkaline, and in alkaline conditions, certain types of kidney stones called struvite stones are more likely to form. So bacterial infection in the urinary tract can, in some cases, contribute to the formation of kidney stones. It is a quiet and somewhat indirect effect, bacteria changing the chemistry of their environment in a way that has consequences for the physical chemistry of mineral crystallization. Life, as always, quietly changing the world around it, gently moving forward. Let us think about the bacteria that live in our eyes. The conjunctival microbiome, the community of bacteria living on the surface of the eye, and the inner surface of the eyelid, is much less diverse than the gut microbiome. But it is present, and potentially important. The eye is a relatively hostile environment for bacteria. It is bathed in tears that contain antimicrobial compounds, including an enzyme called lysozyme that breaks down bacterial cell walls. Despite this, a small number of bacterial species manage to persist on the ocular surface. These resident bacteria may play a role in protecting against pathogenic bacteria trying to colonize the eye. Research on the ocular microbiome is relatively new. The tools to identify and study very small and diverse microbial communities have only become widely available in recent decades. The eye is one of the quieter frontiers of microbiome research. Now, as we continue, very gently now, let us think about bacteria in ancient amber once more, but from a different angle. When insects are preserved in amber, encased in the sticky resin of ancient trees, and then fossilized over millions of years, the bacteria that were present on and inside those insects at the time of entrapment may also be preserved. Some researchers have found evidence of bacterial cells in amber-preserved insects that date back tens of millions of years. Studying these ancient bacteria, comparing them to their modern relatives, offers a window into how bacterial communities associated with insects have changed over evolutionary time. It is another form of the quiet conversation between the present and the ancient past that science makes possible by carefully studying what is preserved in amber, by reading the chemical and structural information trapped in those golden stones. We can hear something of the quiet story of life from an unimaginably distant time. Slowly, gently, let us think about bacteria that can break down rubber. Natural rubber is derived from latex, a milky fluid produced by certain plants. It consists primarily of polyisoprene, a long polymer chain that is quite resistant to biological degradation. And yet certain bacteria, including species of Gordonia and Nocardia, are able to break down natural rubber. They produce enzymes that cleave the long polyisoprene chains into smaller pieces, which the bacteria then use as a carbon and energy source. This ability is rare and specialized. Most bacteria cannot do this, but its existence tells us again about the metabolic reach of the bacterial world. Almost any organic molecule, no matter how stable or tough it seems to us, will find somewhere in the bacterial world an organism that has evolved the enzymatic tools to break it down. The living world has, over billions of years, developed the chemistry to disassemble almost everything it has built. Let us take a quiet moment to think about bacteria and the concept of intelligence, not in any grand or dramatic sense, but in the quietest, most minimal possible sense of the word. Bacteria have no brains, no neurons, no consciousness as we understand it, and yet they process information. They sense their environment. They integrate multiple chemical signals simultaneously. They make what we might reluctantly call decisions about when to form biofilms, about which direction to move, about which genes to turn on or off. There is a growing field of research called bacterial cognition that explores very carefully, and with many caveats, the question of how information is processed. At the bacterial level, the conclusion is not that bacteria are intelligent in any meaningful way, but there is a kind of quiet and beautiful information processing happening at the cellular level, a processing that, at its most fundamental level, is not entirely unlike what happens in nervous systems, just quieter, much, much simpler, and billions of years older. Now, as we come closer to the end of our time together, very gently, let us think about the feeling of lying in grass on a warm day. If you have ever done this, pressed your back gently into a lawn or a meadow, you were in close contact with one of the most richly bacterial environments on Earth. In the soil immediately beneath you, and in the air rising gently from the warm grass, were millions of bacterial cells. The earthy green grassy smell of warm soil and vegetation is in part a bacterial smell, geosmin from actinobacteria, and dozens of other volatile organic compounds, released by the bacteria living in and around the root zone of the grass. The warmth of the sun on your skin was warming the soil too, speeding the metabolism of the bacteria within it, causing them to produce more of those gentle aromatic molecules that rise and mix in the quiet air. Even the most peaceful, ordinary moment of a summer afternoon is quietly full of bacterial life. Slowly, gently, let us think about the bacteria associated with flowers. Flowers produce nectar, rich in sugars, and pollen, which is rich in proteins and fats. Both of these are excellent nutrients for bacteria. Bacterial communities have been found living in the nectar of flowers, in the pollen, and on the petals and leaves. These bacteria interact with the pollinators that visit flowers. Bees and other insects collect pollen, and in doing so, they also transfer bacteria between flowers. Some bacteria found in floral nectar produce compounds that affect the behavior of visiting bees, influencing how long a bee stays on a flower, or whether it returns. The ecology of bacteria in flowers is still being explored and understood, but the image it suggests of flowers visited not just by bees and butterflies, but by invisible communities of bacteria, quietly living in the sweet nectar and drifting gently from flower to flower on the bodies of pollinators, is a very gentle and beautiful one. Let us take a very quiet moment now, close to the end of our journey, to think about what it means that we live in a world so densely inhabited by bacteria, and yet perceive almost none of it. Every breath we take contains bacteria. Every surface we touch carries them. They live in us, on us, and all around us. They are in our food, in our water, in the air, in the soil, in the clouds. They have shaped the planet we live on, created the oxygen we breathe, built the soil we grow food in, cycled the carbon and nitrogen that make life possible. And all of this happens in a realm of scale that is simply invisible to our unaided senses. We are surrounded by a world of extraordinary biological richness and activity, and we move through it in quiet, comfortable unawareness. Not ignorant, exactly. We know it is there. When we think about it, just unaware. In our moment-to-moment experience, there is something very gentle about that thought. And so we come to our final fact, our 120th gentle thought. And we arrive quietly, back where we began. Bacteria are the oldest, most numerous, most widespread, and in many ways, most fundamental form of life on Earth. They were here before us. They shaped the conditions that made us possible. They live within us, and without us, they will outlast us. They are neither our enemies, nor our servants, though they are sometimes harmful, and increasingly we have learned to use them as tools. They are simply life, ancient, quiet, persistent, endlessly varied, endlessly adaptive, endlessly patient life. They do not know that we study them. They do not know that we have spent this time tonight thinking about them. They simply continue in the soil, in the ocean, in your body, in the air above you right now, quietly, slowly, doing what life has always done, growing a little, dividing, waiting, beginning again. And so, we come gently to the end of our time together tonight. We have spent these quiet hours, wandering through one of the most ancient and quietly remarkable worlds that science has given us the ability to explore. The world of bacteria. We thought about their age, billions of years, quietly outlasting almost everything. We thought about their smallness, 1/1,000,000th of a meter, inhabiting a world invisible to our eyes. We thought about their shapes, spheres and rods and spirals and stars, each one a tiny, elegant form. We thought about their movements, flagella spinning like molecular propellers, cells gliding softly across surfaces, twitching forward one small pull at a time. We thought about the oxygen we breathe, which came from them, the soil beneath us, which they built, the food we eat, which they help make, the bodies we inhabit, where billions of them quietly go about their work. We thought about bacteria in ice and fire, in the deep rock, in the clouds, in the ocean, in the roots of plants, in the gut of a bee, in the light organ of a squid in a dark ocean. And across all of it, there was a kind of quietness, a kind of persistence, a kind of gentle, unhurried continuation that has lasted for more than 3 billion years and shows no sign of stopping. There is no need to rush. You can let all of these ideas drift softly through your mind, the way bacteria drift through water, slowly, without any particular direction, just moving gently through the space available to them. The soil outside is full of them right now, quietly working in the dark. The air in this room holds them, gently suspended. Your own body, warm and breathing, is home to trillions of them, quietly going about their ancient, patient work. Slowly, gently, let your breathing slow, just a little more. Let your body feel heavier and softer against whatever surface is holding you right now. Let the thoughts that have been drifting through your mind tonight begin to slow and settle. The world is full of quiet life. It has been full of quiet life for a very long time, and it will continue long after tonight has passed into morning, long after we have both moved on, quietly, patiently, the way bacteria have always continued. There is something deeply comforting about that. If you let it be, you are part of a world that has been alive for 3.5 billion years. And right now, all you need to do is rest. Good night. And if you enjoyed spending this time with these quiet, ancient, and endlessly fascinating ideas, a gentle like or subscribe means a great deal and helps these videos find others who might need a quiet place to rest their mind. Until next time, sleep well.